Thank you very much for joining us. My name is Dan Peizer with Catalent Pharma Solutions, and we're very happy to have you here. We're going to take you on a bit of a guided tour of Catalent's thinking with regards to drug development, dose design, and manufacturing. As the leader in the global leader in drug development design, we feel that we've got innovative approaches that are really backed by the 80 years of experience that we have in doing this. And we're elevating the thinking a little bit, and we're delighted to have you with us to join us on this journey. We're going to start with Julien Meissonnier. Julien is the Vice President of our Science and Technology Group here at Catalent, and he's going to discuss how we approach early stage development with some new thinking and data-driven approach that we think that you'll very much appreciate. So, Julien, thank you very much.
Thank you, Dan. Good afternoon, everybody. So the first presentation from these sessions aims at sharing with you what is the mindset, what is the approach, the structure, and the depth in the science we put behind our offering to help and guide our customers when their journey from drug discovery to clinical phase 1. And during this presentation, we're going to take a very specific angle. We're going to aim also at providing you with recommendation to facilitate the selection of an optimal formulation and dosage form at those early days of product development. The word optimal is important, as it does not mean a complex or an advanced formulation, but it means the most appropriate formulation or dosage form for those stage of development cycle. And it's even beyond that. It's the phase appropriate selection. So most of you, we're going to review now this optimal selection in the context of the challenges that each of us know, face, and endures in the course of product development, encompassing, for example, the prevalence of polywater-soluble drugs, and mainly so-called polywater-soluble drugs. Then afterward, the fact that preclinical models often fail to provide human predictions. The elevated attrition rate also is an important factor, but most importantly, despite the fact that formulation scientists today are more and more skilled at developing complex formulation with advanced delivery technology, oftentimes they're devoting their efforts to try to hit a wrongly defined or a partially defined target. So to avoid those pitfalls, we will provide examples and we'll illustrate one success factor: the integration of drug metabolism and pharmacokinetics along with developability assessments to ensure that we get clear target that development scientists can tackle in their development. So now, in the context of the way we develop a drug product, what is the day-to-day work of the scientists? What are we here tasked to do? So first of all, we're here to assess and select the right molecule form, the right formulation, and the right dosage form. And this by designing the right study, getting the right results, and making the right decisions. So you will guess that this makes a lot of rights in the route to the clinics. And this continuum of making the right decisions follows a stepwise and logical approach that all these steps together predicate successful selection. So a recommended pathway for moving a drug candidate from the late stage of drug discovery to clinical phase 1 take 5 mandatory steps. In the first step, we're aiming at selecting the right molecule, leveraging some DMPK competencies to be able to take the best candidates that has the most potential from a developability standpoint to make it to the first in vivo efficacy assessment. And then we're leveraging easy formulation services, phase appropriate again, that we call Optiform Select, to be able to propel the molecule quickly to those first animal proof of concept study. With this, you got started for getting the right molecule that has some legs for getting good proof of efficacy in animal. The second step is selecting the right molecule form. You get your molecule, you scale it up, and then you need to characterize it. Select the right salt, select the right polymorph. And here, with a finite, but slightly higher amount of API molecule, you need to be able to generate as many body of knowledge of your drug. to be able to risk assess where it needs to be and what's the target for your development. With this, in Catalent, we're going to talk about it. We utilize semi-automated and automated platform so that with this finite amount of API, we can generate the body of knowledge and know our drug better. So this is the right molecule form. Next step is about getting the right formulation concept. So with this, we need to have a toolkit of enabling and conventional technologies to be able to action those based upon the finding we have with the molecule from those first steps. And then ultimately propelling the formulation concepts to the right dosage form, either for GLP toxicity studies or for the clinical phase 1 at the end. So now let's start with the first section, which is about getting the right molecule. At this discrete point in time, we have molecule series of a given family that have proven in vitro efficacy in secondary assay. And the main question is which one has some legs to make it to get in vivo efficacy? How could I propel the best molecule to my first in vivo efficacy model? So this is what we're going to illustrate example where we bring together the drug metabolism and pharmacokinetic assessments along with developability to make the right choice and get the right molecule to the next stage, which then could be formulated with few hundred milligrams to get the best shot in the animal model, with putting us away of any harm and any risk of getting solubility hurdle, permeability hurdles, so we get the best 3 to 4 day formulation services to get you the best chance to deliver your molecule in animal to verify if it has any legs. So we're going to provide one example that's oriented around what's the key question, what's the best molecule. This is a family of 5 compounds that we rank order here by their efficacy. And we're going to see compound 1 ranks the lowest efficacy in secondary in vitro assay, whereas compound 4 ranks the highest one. So how do we select this drug? Based upon efficacy, it's a partial description. So what we've been doing a developability assessment using the developability classification system. We're going to see a bit later in the presentation. But the main conclusions that you need to get from this slide are the following. Among those 5 drugs, 4 will need formulation support. Thus compound 1, 2, 3, and 4, with the exception of compound 5, which is a soluble drug. What does it tell us? Second constatation is the following. Efficacy does not rank with developability. So you need to make some trade-off. It starts to be a bit more complex when you're bringing the way the compound behave in vivo. So we're modeling the same 5 compounds using Berkeley Madonna software and GastroPlus, and we've been assessing what's the percentage bioavailability of the drug. And again, create a second layer of complexity. But the conclusion of this study is when we bring in, and we've been measuring for those 5 compounds, the clearance, so that we can get both the bioavailability and the fraction absorbed. And here are the conclusions from this slide. We get it here. Compound number 1, we see as a fairly moderate efficacy. The higher the score is, the lowest the efficacy is. And we see that it is a polypermeable compound and polysoluble compound. And even with the fraction absorbed, which is 31%, even if you resolve the solubility challenges, we see that 31% goes in 13%. So if you double that, you're not going to get more than 26% of overall bioavailability. So this is driven by high clearance. So this one is to be killed. The second compound is solubility limited, DCS class 2. Solubility is the main hurdle. But we see exposure, absorption translating exposure. There's no clearance. So any slight improvement that you can make with this compound, which is fairly active, in terms of solubility leveraging, will have a significant impact on exposure. So that's the reason why this compound has been selected as a backup compound. And we continue as dose 4, but the one I wanted to highlight is the compound 4, which is the one which had been selected. It's a DCS2A compound, very strong efficacy, rank 1. DCS compound means that the main hurdle to absorption is dissolution rate. You need to go fast within the allowable absorbable window. So it's an easy formulation challenge per se. It's not resolving a true solubility issue, it's resolving the speed at which you get a drug in solutions. And obviously, it has a limited fraction absorbed, but it has also limited bioavailability, so low clearance. So this compound is interesting because it means the formulation scientist can make easy work by increasing the dissolution rate with co-micronization, micronization, or any other associated technologies, so that the fraction absorbed increase and equally important, the exposure increase on the same manner. So this is the important factor. Compound 2 is the lead, compound 4 is the backup. We move with 3 to 4 days formulation services. Here it was in the US, our facility in San Diego. We got a drug that we propelled with compound 2 to the clinics, to the animal studies, and we got exposure. And the next stage was synthesis scale-up, so that we can move to the next stage, which is the pre-formulation study and characterization of the drug candidate. So what we're here to do here is to understand what is the best molecule form. For example, your compound has stability or solubility issue. Can I make a salt as soon as possible? So we have a semi-automated platform where we can run between 48 to 96 salt screening activity with a limited amount of API, roughly in the gram level, and therefore make the right decision on what is the best salts. And at the same moment, on the same semi-automated manner, characterize intimately what are the properties of the molecule, physicochemical properties, to derive from its risk profile. What's the chemical stability risk profile? What's the physical stability risk profile? What are the manufacturability of this drug candidate, so that we can move to the next stage. And with this, we know your drug. You know your molecule, and you can move to the next stage. Next stage is formulation selection. So here, we're going to try to answer a very complex question. There is the need to make a bridge, and this is an important bridge between molecular properties and formulation. And this is not an easy bridge. This always starts with an easy statement. I got a BCS class 2 drug or poorly soluble, or I got a BCS class 1 drug soluble. But what does it tell us? It doesn't tell us anything more than this very nice representation from Gordon Amidon turned into Michelangelo at the occasions of the BCS, biopharmaceutical classification systems. You might recognize it in the shape of the Chapelle 16 ceiling, right? And what does it tell us? Again, little. You are in a BCS class 2 drug, you need to go to the formulation purgatory. Hurrah. You are with a class 1 drug, you are in the even. But that's the best you can do here. But that's the reason why back in 2010, people like Jennifer Dressman, Dave Elder, and James Butler have assessed that the BCS cannot be derived from its primary purpose, being a biowaiver, because it bears 3K in precision. Number one, aqueous solubility in the BCS is a partial descriptor. BCS evaluates solubility in 200 milliliter fluid volume, aqueous media, different pH across physiological range, but we know, and there's many evidence that aqueous solubility does not correlate well with simulated solubility in intestinal fluid from human aspirates, where it had been measured. Second in precision after aqueous solubility is a partial descriptor is what do we make with solubility in relation to the dose? A compound which is soluble in the microgram per milliliter level might be soluble if it has to be delivered in the microgram range. And third, and most importantly, BCS does not factor at all permeability, despite this very nice split in the diagram in 4 quadrants. But indeed, high permeability can balance solubility. When the drugs go in solution in the GI fluid, progressively quickly permeate through the GI wall, reducing the concentration in the lumen and reducing the solubility hurdle. So that's the reason why the DCS had been set forth, revisiting the BCS class 2 universe into 2 areas. DCS2A compound, dissolution rate is the limitation. DCS2B compound, intrinsic solubility the equation. And that's when we said in the introductions, the high prevalence of poorly soluble compound is biased, because it's based upon aqueous solubility, not based upon dose in relation to solubility, and most importantly, we don't factor in the fact that sometimes solubility and dissolution rate is another hurdle. And obviously, this is a different formulation strategy. Once you characterize your molecule, you got a different scenario. You need some enabling technology suited for DCS2A compound, which are particle size reduction, particle engineering, micronization, co-micronization, salt conversion. But if you go with a real intrinsic solubility limitation, you need some more advanced technology that tackle the solubility of the API by making it already molecularly dispersed in a liquid, lipid systems, or in a solid substrate, solid dispersion. And then afterward, those evolve very naturally in the associated dosage form for either GLP tox study or CGMP phase 1 studies. So, once we set the premise for developing the right formulation, what happens? Formulation scientists prototype it against those objectives. They do in vitro assessments, and they run into animal model, which, by the way, have a limited predictability toward what is happening in human. So here, we're raising the questions, how do we bridge this gap between in vitro and in vivo? And how do we set the target from the beginning and by design? So here is a nice example where you have various dissolution profile of various solid dispersions that perform very differently here on the left side. It's in vitro. You would guess that that formulation will have a different performance, but that's what you get in the in vivo model. What does it mean? Is maybe those people have been trying, making those publications, have been trying to hit at a too high target by evolving and understanding what is the DMPK consequence and DMPK challenge of the molecule. They would have known that progressing by 60% the solubility challenge will allow you to getting a lower exposure or not a so steep increase in exposure. And we're going to see an example that leads that, leveraging PBPK modeling from the start as an enabling tool to understand what does solubility enhancing technology can bring as a benefit to your drug in light of its intrinsic PK and PD properties. It's important to put that in context. So the example we're gonna utilize to end up this presentation is the following. This drug is a real drug we work with our team in Europe. It's a DCS2A, DCS2B borderline drug, right? It's a very high dose. This has to be beared in mind. And most importantly, the activity of this molecule is not related to exposure, it's related to the Cmax. You need to hit the plasma concentration the highest possible to reduce the dose, and the target is engaged. So that's what you need to get in mind. So we establish a PBPK model with the two first row column of this table. Reference 1, reference 2, tested. Coarse molecule, mild molecule. We'll build the model, so you see the dots are the real-life data that we get from animals, PK study, and the full line are the DMPK modeling using GastroPlus. So they match the 1/over pretty well. And what we said at the very beginning, before even starting works, we ask ourselves, what can we get if we get an ideal scenario? That means we resolve entirely, by any formulation magic, the solubility hurdle, 100%. It's 100% soluble. This is what you get. You see the Cmax predicted 1,000 nanogram per milliliter move to 1,500, 1.44 ratio increase. That's an important information. You will not by any magic double or divide by 2 or 3-fold the dose you need. You're going to divide by a 1.44 ratio max. So you have to put that in relation with those you're targeting. And we make the same thing with 60% dissolved, 30% dissolved. And you see that the decrease in improvement is not steep. You're moving from 1,500 to 1,400. Even if it's relative and not the absolute data, it shows you that the difference between getting a formulation that's 100% resolving the issue to 30% is that thin. Does it remind you something? Here, you have formulation that in vitro show that solubility is partially resolved or completely resolved, and they do perform approximately the same way in light of Cmax. So what we've been doing, we've been making a very quick parallel screening approach, 12 weeks, and in this case we did 10 weeks even, where we've been developing the best prototype we could in the various solubility enhancement technology, lipid system, hot melt extrusion, spray drying, and micronization. And we rank ordering those molecules by their ability to be stable, their drug load, important because it's a high dose compound, and the percentage increase they provide in this in vitro evaluation model. And then we've been translating it based upon the DMPK model, based upon the curve you get, to the Cmax increase you can get. What it tells us, lipids, pretty nice, it increased the solubility quite well, no precipitation, but at the end of the day, 15% drug load, and the benefit you got is a 1.44 ratio increase. Hot melt extrusion was not stable, so out of the equation, and spray drying doesn't make a steep solubility increase, but has a higher drug load, and Cmax increase of 1.3. So on spray drying, will you drop on 70% your drug load to get a 1.43-fold increase? Where do you get the lowest pill burden for the patient? The patient need is not satisfied. So at the end of the day, we recommended co-micronization approach with an 80% drug load using, we co-micronize the drug with surfactants to elevate its solubility. We did roller compaction We have a key site for doing that in Kansas City in the US and in Schondorf in Europe, where we are mastering this roller compaction process. And the solubility increase was good enough, 35%, which gives a 1.3-fold increase in Cmax. So by this, you got the highest drug load you can get with the technology, and you benefit from a 30% increase in Cmax, which means a 30% decrease in drug load. And here you got the best that you can, and you got a phase-appropriate model. And then once the formulation concept is selected, it's quite easy to propel that to the dose form. That's suited for either GLP tox formulation, giving it to an animal at high milligram per kilogram body weight, for example, a dispersion in CMC solutions, in CMC liquid systems, as we did it for this specific drug. And for the end stage, a powder in capsules approach or a spray-dried roller compacted product will be good for phase 1. So you see the importance of getting a toolkit that's complete so that you do not have any disruption in the model. So to conclude, and being mindful of your time, in summary, this presentation and these discussions highlight the importance of getting a continuum of services ranging from candidate selection, molecule selection, pre-formulation, formulation selection, GLP toxicity formulation, and cGMP phase 1 formulation capability. This is the toolkit. Then this continuum of services is supported and should be supported, that's our own view, by the progressive and the accurate setting of a drug, metabolism, and pharmacokinetic model, so that scientists' effort could be targeted at hitting the right target and can be efficient. And this is the enabler. But most importantly, those processes must be supported by product development teams that are fast, reacting fast, that are flexible, so that they can handle any new data they're going to generate and leverage their toolkit on a fit-for-purpose basis, so that you can react to real-life expectations that are happening. And that's what Catalent had been good at. We had operated over the past 8 years, hundreds of molecules, and now we feel and we sense that bringing this very convergent continuum of services is and will continue to provide value to our partners and to the patients. So with this, I wanted to acknowledge and recognize the work done by your scientific advisor in the field of drug metabolism and pharmacokinetic, Dr. Jan Nelissen. If you're interested in learning more how we put DMPK along with developability assessments at the forefront and the core of our product development process, I would invite you to attend this webinar on November 28th. And should you have any questions, there's going to be a Q&A session at the end of this session. Or also you can join us at Catalent booth on the Hall 4.1. Thank you for your attention.